We're very grateful for you joining our first reproduction strategic research initiative showcase. So here we wanted to highlight the work across disciplines. It's our we're now in our third year since launching the SRI, and we were awarded recently a 3-year renewal, and the comments were extremely positive about the SRI. I think they were incredibly impressed by the breadth and depth of expertise. So I'm really looking forward to today's talks. We have a number of early career researcher chairs. And so thank you in advance also to the speakers for representing all of the, at least as many of the disciplines that we could represent in a short showcase, but by no means all of them. But yeah, thank you very much. And if you have any questions, please feel free to use the chat box or the raise hand feature. And yeah, looking forward to the discussions.
Thank you for the introduction, Kathy. So just to let you know a bit about the format, we're going to have 4 presentations for the first session. We've asked our speakers to limit themselves to 5 minutes, so these should be flash talks. And then a joint Q&A at the end of around 10 to 15 minutes before moving on to the next session. Please keep your microphones muted throughout the flash talks. And for questions, please do put them in the chat or use the raise hand function on the bottom right of your screen to ask them at the end during the joint Q&A. And without further ado, we'll move on to our first speaker for the day. It's Dr. Geoffrey Maguire from the Faculty of Modern and Medieval Languages and Linguistics, who will be talking about queer conceptions and interdisciplinary approach.
Thank you very much for that. I probably should start out by saying I don't usually dress like this for presentations, but I've just come from a graduation, and I'll be going to another one in about 25 minutes or so. So I'm afraid I can't stay for the discussion afterwards, but I do very much look forward to catching up on YouTube next week. So the presentation that I hope you can see. Yep, that I want to introduce to you today is called Queer Conceptions. And it's a research network that Francesca Gaccioli and I set up last year or the year before in collaboration with the Reproduction SRI through their conceptions funding. And as you'll see in the title here, I've included the term interdisciplinary because I think, well, really, I mean, I think that type of word has become, I think, slightly perhaps overused in the academy over the recent years. But I really think that this is a project that does, at its very heart, have an interdisciplinary ambition. So, you know, Francesca, the co-lead, is from the Department of Obstetrics and Gynecology, and I work in film studies in the Faculty of Modern Medieval Languages. So 2 disciplines really that conventionally and traditionally are miles apart. But what I think we do share is a focus from our distinct disciplinary and also personal perspectives on reproduction and on the changing composition of families in the 21st century. So from my point of view, for example, in film studies, one of the things I've noticed over the past 10 years or so is a real upsurge in cinematic portrayals of queer families, you know, which come very often as a result of changes in legislature, for example, in particular countries. And I think documentary cinema in particular really is at the forefront of capturing some of these, you know, pretty seismic societal and legal changes. And you can see some examples here of documentaries about same-gender parenting, adoption and surrogacy, also some documentaries about activism. So there's a very quite a famous one here from Ireland over the last few years, which did very well on the festival circuit about equality laws, and particularly about adoption laws in Ireland. Documentaries about queer childhoods, but also really interestingly, I think, quite a few films over the last 5 years or so about trans pregnancies. And what I think these films do very well, of course, is kind of demonstrate and show the joy of how many more people than ever before can start families thanks to the advances in medical, in medical science and the reproductive sciences, and thanks to new legal routes for adoption and surrogacy. But what I think they also do is highlight the problems and the difficulties for LGBTQI people in particular, given that legal frameworks, access to medical care, societal acceptance and tolerance across the world differs, really quite radically. And one of the things that Francesca and I really wanted to do through the Queer Conceptions Research Network was to begin to facilitate public dialogue and discussion about queer routes to conception. Because for us, at least, it seemed that debates in the UK had evolved really around two distinct poles. So the clinical and scientific possibilities for queer reproduction were very often completely removed from the actual lived experience of going through treatment itself. So for the purposes of today, in my 5 minutes, I just want to talk very briefly about one event that we held a couple of months ago at the Cambridge Science Festival, when we screened the brilliant film Seahorse, which tells the story of Freddie McConnell, a trans dad who the film describes as the dad who gave birth. And if I had lots more time, I would tell you about all the ways in which COVID completely and utterly annihilated every single one of the plans that we had for the network. But I will not go over that, but I will say that one of the really unexpected, the unexpected advantages of moving everything online was that we actually had over 400 people sign up to attend this screening, which led to a really interesting debate afterwards at the follow-up panel that we had. And on that panel, we had Freddie, the dad, in discussion with various experts on reproductive rights from across the university. So from sociology, from the medical school, from the Centre of Family Research, and from ReproSoc. I can actually see some of you in the audience today, so thanks again. And what really, what emerged in that discussion, I think, and from the questions that the public were asking afterwards was a real desire to bridge this gap between scientific discourse and then also the lived realities of trans and queer parenthoods. And I think what this film did very sensitively was depict some of these problems and some of these difficulties, whether that be Freddie's repeated misgendering by hospital staff, the forms that he had to fill in, which didn't allow him to register his gender, the huge psychological impact, in fact, and physical impact of having to stop hormone treatment, testosterone treatment, to then undergo IVF. And also the legal fight that still continues to be officially registered as the child's father rather than the child's mother. And how am I doing for time? Yeah, I'll jump to this. But the feedback that we received, I think, what it really demonstrated, first of all, is that there's a huge appetite for discussions such as these. And what the feedback showed us was there was a real appreciation for the visibility of issues related to queer and trans routes to parenthood, on such a public platform. I think one of the, I'll skip through this slide just to say that some of the most interesting feedback was from medical practitioners, and we had a couple of midwives there who really for the first time I think had the opportunity to think about the distinct difficulties of going through these already difficult pathways to parenthood, but from a perspective of trans and queer people. And really the feedback gave us two quite interesting things to focus on. First and foremost, what happens after pregnancy for these children from diverse families, particularly with early school education and primary education. And then also to think a bit more clearly about, to think intersectionally about queer and trans people of color and how very often these problems are compounded, particularly societal problems. So I realize I'm out of time, so I'll leave it there and just say a huge thanks to the Reproduction SRI for allowing us to get going with these events that we've had over the past year and a half or so, and particular thanks to Christina for what was quite an administratively turbulent period over the last year or so. And please do get in touch if you have any suggestions for events or if you'd like to be involved. Thanks.
Well, hi, everyone. Thanks so much for having me. And thank you to Dr. Maguire for that wonderful talk. My name is Grace Kromm, and as you said, I'm from clinical neurosciences, and I'm a current PhD student. Today, I'm going to be speaking to you a little bit about the research study that I'm setting up for my PhD, which is called Brain Activation in Mother and Baby, or BAMBY. And here you can see some of the people associated with the project. And right now we're really just setting up and piloting, part of which is very graciously funded by the SRI Incubator Fund. So I'm going to briefly introduce you mostly to the plans for our project. And what BAMBY is primarily looking at is something called neural synchrony. And at a basic level, what neural synchrony is the synchronization or coordination of brain activity during a real-time interaction. So that can be correlational. So when A is high, B is high, or directional. So that's changes in A forward predict changes in B. And so this is these are some of the ways in which papers depict neural synchrony. And neural synchrony has been measured in quite a few contexts already. We know that it's higher between romantic partners than between strangers. And we know that neural synchrony is higher when pairs are cooperative rather than competitive. And we know that there are cues like eye contact that it can increase this neural synchrony. And a few studies have already looked at neural synchrony between mothers and their older infants. And they found, for example, that positive emotional affect, so really just smiling or looking happy, already elicits increased neural synchrony between the dyad. And so the theorized functional significance of this neural synchrony is that it coordinates neural oscillations in the infant's brain so that entrains the baby's brain to the patterns of social communication, teaching the baby's brain how to receive the social information, respond appropriately, and also promoting mutual receptivity that really keeps the dyad quite literally in sync. And our project is looking to measure neural synchrony for the first time in a neonatal context. And so we're going to be doing this with dual EEG, and that's the technique that you see here. And so when my PIs and I were first thinking about how we might want to measure this neural synchrony with newborn babies, it made sense to think about, well, what are the main ways in which parents or mothers communicate with their newborn babies when, they could be physically separated for the first time? And the natural answer was communication through affectionate touch. And as many of you may know, affectionate touch has an extremely important role in early bonding, in decreasing long-term stress or cortisol reactivity, particularly through the hormone oxytocin. And affectionate touch is also essential for baby neurodevelopment. And there is an anatomically segregated pathway for affectionate touch, and that's through something called CT fibers, C-tactile fibers in the skin. And they're optimally activated by gentle stroking at a very specific speed, 3 centimeters per second. And they activate effective processing areas in the baby's brain, such as these, or in all brains. And so overall, our design is looking at changes in neural synchrony across different types of affectionate touch. And this is in the days right after birth. And so to do this, we're manipulating two things. First, the sociality of the touch. That's whether the touch is delivered by the mom herself, so that's social, or whether the touch is delivered without the mom, and that's sham. And second, we're manipulating touch mechanism. So that would be whether the touch optimally, suboptimally, or minimally activates those CT fibers that I just told you about. And that's dependent on the speed of the stroking, again, which will be across the baby's back. And so that design yields 6 touch conditions, which you see here. And our hypothesis is that both sociality and touch mechanism will matter for neural synchrony. So we would see maximal neural synchrony in this top condition here. And we're also interested in looking at individual differences in this neural synchrony, what might be associated with this neural synchrony in moms or infants. And so we're looking at, for example, how average neural synchrony might relate to oxytocin levels in the mom or baby, or cortisol levels in the mom or baby. And so we're going to be measuring those with saliva samples from both. And we're also going to be looking at behavioral subscores in the babies, particularly interested in social interactive capacities, self-regulative abilities, and that we're measuring with the NBAS, the Neonatal Behavioral Assessment Scale. And so in all, just to summarize, while we know that neonatal affectionate touch is extremely important, we don't really know how moms and babies' brains might behave and engage across different types of affectionate touch. So by measuring this, we're really hoping to provide insight into the neural importance of affectionate touch for newborns, as well as the neural basis of maternal newborn communication in general. And we're just hoping to inform how we might be able to protect and promote healthy development through touch. So that's all I have. I wanted to keep it under 5 minutes. Hope that was a good amount of time. Thank you all so much for listening, and thank you to the SRI for hosting this showcase.
Okay, so while Christina's getting the slides up, I'll just explain that I'm giving this talk, this flash talk, on behalf of my PhD student, Helen Dolling, who is on compassionate leave. This is very much her work. And it's work that also is embedded within the Next Generation Children study, which I think got referred to in this morning's meeting as well. The context for this work was the kind of diagnostic odyssey that many parents face, that the problem is that there's about 300 million people in the world who have an undiagnosed genetic condition. So that's more than cancer and AIDS put together. And it can take between 6 and 8 years for a genetic diagnosis to be found, or at least it was historically. And obviously that's an awful lot of uncertainty and stress for people. And so the Next Generation study is the first large-scale study to involve rapid whole genome sequencing, so rapid genetic testing for the trio, so mum, dad, and child, and that has a whole lot of positives, because what used to take 6 years is now, that information can be available in as little as 6 days. So there's huge amounts of potential from this. I mean, we all know that the saying, knowledge is power, and the view that diagnoses, well, they don't just reduce uncertainty and therefore stress, but they can guide treatment. They might give you access to resources. So there's lots of ways in which having a diagnosis can be a really, really positive thing. Anyway, so I was talking about some of the positives, but clearly there are dangers as well. So in the field of neurodiversity, a metaphor that sometimes people use is that you think you're traveling to Rome and you end up traveling to Amsterdam. And Amsterdam is a lovely place, but if you thought you were traveling to Rome, then it can take an awful lot of time to adjust to the fact that it's not quite as sunny and all of that. Now it's quite a rosy metaphor, that, because actually what life is like with a healthy child is completely different from what life is like with a sick child. So that transition is already very, very stressful. If on top of that, you're suddenly being given an awful lot of unexpected information that might be quite complicated and difficult to understand, it could be that parents feel overwhelmed, that that's too much information, it's too soon. And it may not actually, it may be something that completely changes your identity as a parent. You no longer think of yourself as a parent, but as somebody who's caring for a sick child. And perhaps that diagnostic information doesn't actually come with any potential for treatments. So there are dangers as well. So the idea is that by getting parents' views, we can really get a much better understanding of the perils and pitfalls of this journey, if you like. So the important thing to say here is that Helen Dolling has for some years been the research coordinator on the Next Generation Children's Study. And so she's met all of these parents at their time of crisis when their baby was very ill in neonatal intensive care and getting treatment. So she has a rapport with them that is really unique and special. So she's going to be conducting interviews that might take an hour, might take more than an hour. And the focus of these interviews will be to look at what they think about the timing. When is the best time to get this test? Is it that, you know, the sooner the better, or would they like a period of kind of adjusting first? And then having had that diagnosis, or having the testing and that not actually resulting in a diagnosis, because actually it's only just over a third of those tested where they do get a concrete diagnosis. Did they find that process helpful or harmful? And importantly, is there any kind of element of decision or regret? Do they wish, would they like to turn back the clock? Would they rather they didn't know that testing hadn't been done? So that's the goal of it. So this is just to show her design. She's going for 90 families where they've either got a diagnosis or not, and whether that diagnosis was early or not. Next slide. So that's the second goal. Within that 90, her aim is to explore the variability, because of course we don't expect the answer to be the same for all families. And then if you move on to the very last slide, bringing fathers into the picture, we've had a nice little incubator fund from the Reproduction SRI, which will allow an additional 30 interviews to be done with dads, so that we can get the father's perspectives in there as well. Because while to mother and to father might sound like very different things, actually today the experiences and the viewpoints are often strikingly similar. So it would be a real omission to miss dad's views out of all this.
So I'm representing the Cambridge Family Law Centre at the moment. So the name indicates what we do. And obviously we're also looking at parent-child relationships, and indeed the person chairing this panel is one of the main players in this at the moment. So it also has the name law in it, and I always feel a bit intimidated in company as yours, because we all know that lawyers are a bit silly, they're a bit limited. They're not very creative, and they're not very good at sort of taking things on board that don't fall within their learned patterns. So, you know, the starting point for most of these things was, oh, every child has one mother and one father, right? And of course, that's never been right in the first place. And that, of course, is one of the issues that the law has, not least because in recent times we've faced many societal changes. So same-gender parenting, rainbow families, men giving birth indeed, and multi-parents and single-parent families, which we should forget about. And that at the same time is combined with significant medical advances, assisted reproduction, including surrogacy, gamete donation, mitochondria donation, uterus donation. So we're looking at a brave new world here. And the law is not set up particularly well to deal with it. The laws, many jurisdictions, fail to accommodate the lived realities of children. They don't deal with modern family forms. And I always hate that term because, you know, they're not really modern. They've been around for a long time. They certainly do not reflect all parental ties or parental links that a child may have. And therefore, what I've been looking at, and indeed what Dafni will be looking at, is sort of a radical rethink of the way we define parent-child relationships. And perhaps actually it's not so radical after all if we're looking at it in more detail. So the idea is to break the two-person paradigm, the idea that there is only one mother and one father, and that's that. And to actually look at parent-child relations in a more sophisticated way. And for that, building on the work of my colleague Andrew Bainham, I have been suggesting that we should split what normally the law perceives as a parent into three very different levels. And the English language allows me to do that. If I tried that in German or Danish or French, I would fail, or Hebrew, and all of these. So, and the terms I will be using is parentage, parenthood, and parental responsibility, hence the name of this short presentation. So the short version is that parentage essentially is denominating the biological relation an adult has with the child. And I very deliberately choose the word biological because I do not think this needs to be restricted to genetic links, which of course would include donors of mitochondria, but for example, the surrogate would, in my understanding, also fall under this connection because of the physical, biological contributions of this child coming to this world. Parenthood, then, on the other hand, is denotes a person who by law is declared to be the parent of a child and with whom legal kinship is established, and that of course means legal relations with the wider family as well. So that could be a couple who conceive through artificial reproductive techniques, although the gametes are not theirs, or adoptive parents, where of course there also is no biological link as such. And the final, but by no least important stage is the one that I call parent responsibility. It's in many other countries called something else, guardianship, custody, if you so will. I think parental responsibility captures this really nicely because it's not parent responsibility, it's parental. So you don't have to be the legal parent of the child. And also it is all about responsibility and not your own rights. And the term I use for those who are social parents in a way that they need to be permitted to exercise some, many, or all of the functions traditionally associated with being a parent. So to take decisions on behalf of the child and so on. So it's essentially those who have a parental function with regard to the child. And the problem is that in the law of many countries, these three levels are actually not distinguished. They're usually linked by a limited understanding of what a parent indeed parent is. And that's what I call the two-parent paradigm, because that's where it starts and ends for most people. And that exactly is what needs to be broken. Our understanding, the traditional legal understanding of the parental function. Interestingly, parentage is hardly ever recognized in any jurisdictions as a specific legal category. It's usually subsumed into parenthood. And distinctions between parenthood and parent responsibility are actually much more common. But it's actually really very important to recognize parentage, not least from the perspective of the child and the right to know their origins. And so if we don't keep accurate information on this, then children will never be able to find out what their genetic origins, what their biological origins are. And we do know that children, young adults, and indeed adults often do want to know about where they come from. In case of surrogate children, who carried them and why. And not least because if you reach my age, you will be faced with questions like, is there a history of bowel cancer in your family? And you will want to give a proper answer to that. And you can only do that if you actually know the genetic links that you may have to other people, because they're not really asking about your adoptive parents. Okay? So this is what they need to know. As I said, parenthood is the legal position, and that is, of course, at the core. In many jurisdictions, unfortunately, parenthood and parent responsibility are completely linked. So you cannot have a distinction between the two. And since the number of parents is limited to 2, that also means that certain family constellations cannot be accommodated. And I think that is fundamentally flawed, because it just simply does not reflect the lived reality of many children. Children, thankfully, are not lawyers. They just call persons who they socially perceive to be their parents mommy or daddy. And they don't count in 2s, thankfully, either. And the feelings that these children have, the connections that they have, they're not limited to 2 persons. So that means we can actually, on each of these levels, well go beyond the paradigm of 2 and accommodate that in that way the modern family forms. Now, one last sentence. Because the interesting thing about this, legally speaking, is just because you have these legal categories, doesn't mean you have to attach any legal consequences. So it's actually each legal system, each jurisdiction would be free to attach the legal consequences to any of these levels, if any, as they would like. And I have been nudged to come to a close, and so here we are. Thank you.
And thank you to all our wonderful speakers. Please feel free to put further questions and answers in the chat, as Christina has said. And with that, I'll hand on over to Dr. Kristen Belton from the Babraham Institute, who will be chairing the next session.
Hello, yes. Well, it's a pleasure to be chairing this session, and this is my first sort of introduction to the reproduction SRI. So I'm a new member, so it's good to see everyone, even on Zoom. And I'd just like to remind everyone, so the format for this session is the same as the last one. So there'll be 4 5-minute long talks, and we will try and keep the time, followed by a longer session of Q&A. I'd also like to remind everyone, looks like everyone's done it, but just to keep their microphones muted, so there isn't any background noise while people are talking. So I'd like to hand over to Professor Nick Hopwood of the Department for History and Philosophy of Science, who will start us off today. So over to you.
Great, thanks very much, Christian. I suppose first thing to say is welcome to the SRI. Thank you all for coming. As many of you know, the first so-called test tube baby, Louise Brown, was delivered following in vitro fertilization in Oldham near Manchester on the 25th of July 1978. Over 8 million children have been born by IVF since then, and it's the platform for much reproductive biomedicine and human embryology today. But Brown was far from the first test tube baby to be announced, and was not universally accepted as one at the time. Here, I've contextualized her birth with some earlier claims. Since the 1940s, various researchers had reported fertilizing human eggs to produce embryos and even infants. Journalists had written of test tube babies as they anticipated brave new worlds. Now, if you look at the claim column, you'll notice that some later announcements claimed less than earlier ones. So Robert Edwards and colleagues got a Nature article for early stages of fertilization in 1969, when others had already published embryos in the 1940s and 50s. Other announcements claimed quite a bit more, such as 28 children in 1964. That's a tip-off that criteria of success were changing and contested. Now, I could say more about that if you like, but I want to focus on the column for where the claim was staked. You'll see that some were made in scientific journals, some in newspapers and on television, many in both. And you might think it's simple. The real claims had journal articles to back them up, and the dodgy ones didn't. And there is something in that. But for a long time, baby Louise didn't have a journal article either. She was a major global news story, and within hours of her arrival, the Daily Mail announced, "First test tube baby is born and medical history made as a mother's dream comes true." But in a note in the Lancet 3 weeks later, little was given away, and there was no detailed article for over 2 years. The main disclosure was at a symposium at the Royal College of Obstetricians and Gynecologists in January 1979, and in a few other talks. Brown's birth is nevertheless recognized as the founding achievement of reproductive biomedicine, and rightly so. To understand why, we need to consider the full web of communication, as Bruce Lowenstein has called it, the constant interplay between nature and newspapers, television and press conferences, symposia and magazines. Lowenstein reconstructed this web by following the controversy over cold fusion. That was an ultimately discredited claim at a press conference in 1989 that a simple apparatus, a benchtop apparatus, could release huge amounts of nuclear energy. Lowenstein noticed things like scientists studying videotape of television news to learn how to replicate the alleged breakthrough. But in studying this web, he collected material for just a few months and had access in real time. To take this approach to the history of IVF, we need to tackle something bigger and harder to recover how the web changed over several decades between the 1940s and the 1970s. That's a lot of work because historical research has hardly touched the huge media coverage of IVF. The dominant accounts from scientists focus on journals, while presenting the mass media as a hindrance, if perhaps necessary, to prepare public opinion. Scholars who started to look at the press and television have been interested in recovering attitudes, which is fine, of course, but it's not enough. To grasp the operation of the web, it's necessary also to investigate processes. And we need to look at journal science and media science at the same time, but without flattening everything. Now, I've started to do this work, and I'm fortunate to have funding from the Leverhulme Trust, which will let me finish researching and write a book, The Many Births of the Test Tube Baby, about claims to human in vitro fertilization since the 1930s. I have two main questions about this web of communication. How did changes in the 1960s and 70s reshape expert claims? I'm keen to explore especially how the operation of the web changed with greater media involvement in science. A signal of that change is that this is when commentators first wrote about science and the media. It's also a time when journals were threatened by competition from newspapers and by journalists' loss of deference. So my second question is, how did scientific journals keep their status? Journals weren't always the main means of reporting discoveries, but their status had been consolidated after World War II. Now, one line of defense when this was threatened were embargoes, such as the Ingelfinger rule, the refusal to consider a manuscript submitted or reported elsewhere. So by pursuing expert debate into and out of the journals, I hope to go beyond these and show how journals kept their roles, at least in biomedical science. Now, there's more in the project, but it's time for me to stop. So I'm going to hand back to Christian. Thanks very much.
Thank you very much for giving me the opportunity to give a talk here today. So I'm going to be presenting to you a project that's a collaboration between myself and Vicente Perez Garcia. We're both next generation fellows in the Center for Trophoblast Research, and this project's funded by the SRI Incubator Fund. So this is entitled The Search for an In Vitro System to Study Epigenetics in Early Human Placentation. So epigenetics are the modification to DNA itself or its associated proteins to enable regions of the genome to be either open and accessible and actively expressed, or closed and repressed and generally inactive. And this is really critical to establishing gene expression profiles between different cell types. And so if we look across cell types in the human body, what we can see is that each contains a unique epigenetic pattern. And we know that during development, epigenetic patterning is really critical to establishing this cell identity. And this really begins with a dramatic epigenetic reprogramming event in early embryogenesis, where we see that the epigenetic marks that are inherited from the germ cells are progressively erased in the pre-implantation embryo and then reestablished during lineage specification upon implantation. And this process has been really quite well studied in the embryo, in part through the development of in vitro culture methods to be able to grow embryonic stem cells in various conditions that recapitulate stages within this reprogramming. And this, whereas comparatively, we know relatively little about this epigenetic reprogramming event in placental trophoblast. And the placental trophoblast as the maternal fetal interface in pregnancy is really critical for the supporting the growth and development of the baby. And so this highlights a critical knowledge gap that we need to address to be able to fully understand healthy pregnancy. So until recently, we really haven't had the tools to be able to study this critical time point in vitro. But excitingly, in 2018, Okai and co-authors developed a in vitro system where they were able to culture what they've termed trophoblast stem cells that recapitulate this sort of multipotent trophoblast stem cell population, which can then go on to develop the placenta. So critically, they were able to show that they could take these cells and differentiate them into all of the cell types that form the mature placenta in human pregnancy. But what we can see from early characterization of the epigenetic landscape within these cells is that these don't entirely recapitulate the patterns that we see in human pregnancy. And here I'm showing you just an example of epigenetic data, with the top showing you an example of what DNA methylation looks like in first trimester cytotrophoblast cells from human pregnancy. And below, I'm showing you the profile for cultured human trophoblast stem cells derived by Okai et al. And what you can see is that there's a dramatic shift in this epigenetic mark so that it no longer resembles this in vivo pattern. So Vicent and I have proposed to initiate this pilot project to optimize these culture conditions to be able to better study these processes. So our hypothesis is that culturing these trophoblast stem cells as organoids or in 3D will better recapitulate this sort of in vivo methylation pattern of the placenta. And a lot of the basis for this is really derived from cancer research, where it's been shown that if cancer cells are grown in 3D as organoids, they better match the epigenetic pattern of cancer cells that are seen in patients. So we've really begun this project with a very simple study design. So we're taking these human trophoblast stem cells from Okai et al. And growing these in 3 different conditions. So first as organoids, using the culture conditions developed by Margarita Turco's lab here in Cambridge. And then comparing these two trophoblast stem cells grown in 2D, either grown in the organoid medium, because this medium is unique to the typical media used for growing these in 2 dimension. And then we're going to generate bisulfite sequencing libraries from each of these culture conditions to comparatively look at the DNA methylation profiles initially, but with hopes to move on to other epigenetic marks after, to identify the optimal conditions to recapitulate the epigenetic marks we see in the human placenta. So the idea is to be able to then use this tool to study these epigenetic programming events in human placentation, but then also to be able to study their function in human placentation. So thank you very much, and I'm happy to take questions.
Well, yeah, thanks again for the invitation to participate in today's meeting. It's been an excellent session listening to all the different talks. And Courtney beautifully presented what epigenetics is in her talk. And so because this is a broad audience, I thought instead I would sort of focus on highlighting some of the key concepts that we're thinking about at the minute and really try to emphasize what epigenetics might be able to tell us about development and reproduction. And so 2 of the concepts I wanted to highlight in the next 4 minutes or so is related to that of epigenetic memory and epigenetic priming. And these are areas in which we're very excited about at the minute and where we've been able to make some contributions to the field. And I should just add that today I'm talking on behalf of the whole epigenetics program at Babraham. So the work I'm going to highlight is really collective work from the program. And so this will be a familiar schematic to many of you. This highlights the first week or so of human development. And the first week presents some really interesting challenges for how we regulate different epigenetic marks in our genomes. In humans, there's a prolonged period of developmental plasticity. It's really much longer than in other species. And so how cells maintain this sort of plastic, highly regulative state is still really unknown in terms of the basis for epigenetic and genetic regulation. But once this period of developmental plasticity begins to tailor off, cell fate is then specified and the different lineages are then locked in place. And then once the cell fate decisions have been made, it's really important that the cells then don't sort of wobble around and turn into one of the alternative lineage fates. And so there needs to be some memory, some epigenetic memory of what that cell type is. And it's really some of these key ideas that we study quite a bit in the program. And so one way that we've been looking at this is if you take, for instance, an early unspecified human epiblast cell, the cells have different lineage fate decisions to make, whether either they continue to become epiblast or whether they become fated to become trophectoderm, for instance. And then in recent work from my group, we've actually identified the first epigenetic-based barrier that reinforces this division between the different these 2 different cell lineages, such that if you disrupt this particular epigenetic barrier, it progressively leads to an erosion of epigenetic memory. And now we see the cells drifting between the different cell lineage fates. And interestingly, when we disrupt this epigenetic memory in this particular context, it's not a sudden flip in cell fate, but rather it seems to be increasing the probability at which the epiblast becomes misspecified into a trophectoderm cell. And of course, we manipulate this in the context of an experiment in the laboratory, but we know that epigenetic pathways are particularly sensitive to environmental effects during this early developmental window. And so if these environmental effects can disrupt some of these epigenetic pathways, then you can imagine in the developing embryo itself, it may lead to misallocation of the correct lineage fates and causing problems with further development. And the second concept that I just want to touch on today is that of epigenetic priming. And this is really a way that the cell uses in which to keep in this fairly plastic state during early development, but yet sort of poised to respond to appropriate cues when they come along, so that the cells can quickly turn into one of several different cell fates. And so you might liken it to a train waiting at the station with its engine on and just holding until that green signal comes, allowing it to release from the station. And it's thought that once the cell is then moving forward in development, it then has to go through a series of decisions in order to progressively establish different cell fates. And it's thought that each of these decisions are taken rather independently. And actually, what we've recently found from work carried out in our epigenetics program is that actually cell fate seems to be primed in a very specific way, and quite an unexpected way early in development. So that would be akin to the train continuing down the track, and actually all the junctions on the path of that train, all the points would be pointing towards one particular destination. And actually, if you want to change the destination of that train, you have to actively then switch those points at those junctions in order to divert the cell to somewhere else. And by developing some quite sophisticated molecular assays, which can read out multiple layers of epigenetic information in the same cell, you can begin to put together all these different layers in order to build up a very rich picture of how the cells control this switching of these different points in the track. And rather excitingly, what was discovered is that early in development, cells are actually primed to differentiate into a lineage called ectoderm. So these ectoderm cells lead to our nervous system and skin and certain other organs. And actually, if you leave the cells unperturbed, these undifferentiated cells will then almost by default turn into these ectoderm-like cells. And instead, during development, you have to actively flick the switch to divert the cells towards other cell fates. And so here there's an important decision to make, whereby the cells are then diverted, if they receive the right signaling cues, towards a different cell fate. And again, now the tracks must be switched to further specify cell fates. And so how do this so-called sort of priming mechanism work? Well, again, work from several laboratories in our program have identified that there are so-called priming transcription factors, which sit at various points on the DNA and recruit both activators and repressors in order to establish a sort of dynamic equilibrium that somehow achieves a balance. And at the moment, we don't really understand how these activators and repressors are recruited and how this balance is achieved. But then by keeping these sets of genes in this sort of balanced equilibrium, when the cells then receive an appropriate signaling cue, then the appropriate response can happen, whereby activators will take over, leading to the timely induction of these particular genes. However, if you remove the priming factor early on, this disrupts this equilibrium. And frequently what happens is there will then be a sort of over-distribution of these repressor proteins, which means that when the same signaling cue then comes and encounters the cell, actually now the wrong thing basically happens. And here the repressors sit up on the DNA, leading to a failure to activate those cells, those genes in a timely fashion. And one of the ways in which we're studying it in human development is that we've adopted what I think is a really nice, exciting model that was originally published by Naomi Morris and Afonso Martinez Arias in Cambridge. And here they can generate a sort of a 3D aggregate of cells that in many ways can recapitulate aspects of a human embryo that's approximately 2.5 weeks post-fertilization. And as shown here by looking at the signal of a gene called brachyury, you can see that these aggregates spontaneously self-organize and establish anterior-posterior patterning such that you can distinguish the different polarities of this particular structure. And what we find unexpectedly is if we disrupt different priming factors, actually we have hints that this polarity that's being established becomes disrupted. And so this excitingly now links the role of priming transcription factors with establishing the body plan during early human development. And this is something we're very interested in looking at further. So with that, I'll just stop there. And this is people in the epigenetics program and our contact details. And I look forward to any questions. Okay, thank you.
And we start with Professor Steve Charnock-Jones from the Department of Obstetrics and Gynecology, who will tell us about the development of new methods to screen for fetal growth restriction.
I'm really a stand-in for this talk. So Professor Gordon Smith would have been giving this talk, but he's somewhere else. So for those of you who are old enough, you'll know that it's not Smith, but Jones. Alas, not Smith, but Jones. That works for some of you. If you're too young, don't worry. So why, what, and how for fetal growth restriction. So the why. So in the UK, about 1 in every 250 pregnancies ends in a stillbirth. And that's a pretty shocking statistic that most people don't know. So relevance here, growth restricted fetuses are at increased risk of stillbirth. And of those stillbirths, about 30% occur at term, and delivery of a normally formed fetus at term is pretty safe and occurs very commonly. So the overall plan for this work is to be able to develop a screen for fetal growth restriction in late pregnancy and then deliver those babies at risk of stillbirth. And if the baby's delivered alive, then you've saved the baby's life. So that's a pretty dramatic outcome and would be a game changer. But there are challenges with that, as you might expect. Right, slide. Is it working? Is it moving? Yes. Okay, so we've done the why. What are we going to do? Or what are we doing? So the key is to have a good screening test, which has high specificity and high sensitivity. So what you want is something that's good at identifying those who really are at risk and doesn't miss them. But equally, you don't want to falsely identify those who are not at risk, because then you'd end up delivering them needlessly. And we're talking about large numbers here. So that would be a detrimental thing to do. Okay, so how? Relatively simple in theory. So collect blood samples from pregnant women before you know the outcome, characterize the women and work out who ended up delivering a baby with growth restriction. And then go back to the blood samples that you already collected and you've stored and measure something, and it could be a range of things, measure something in the blood, and then compare the cases and the controls. And from that, you hopefully will be able to identify a screening test, which ideally, or maybe actually essentially, you would validate and learn, hopefully demonstrate that your test will work in other populations, in other settings. So this is what we've done. And this is based on a study which has been running in the Rosie for quite a lot of years now. It's the Pregnancy Outcome Prediction Study, or POPS. And it's pretty simple in design, although complex in execution. So we collect blood from women at around 12 weeks gestation, and then again at 20, 28, and 36 weeks. And in POPS 1, we recruited just over 4,500 women, 4,512 to be precise. So they all generate 4 blood samples, which are all in the freezer. So that's thousands, hundreds of thousands of tubes actually, because it's many aliquots. And then having collected that, we identify from that group 175 cases of term FGR and 299 controls, a random sample of normal controls. And then we perform metabolomics, which is essentially a way of measuring, in this instance, 837 named metabolites in those blood samples from those women at those 4 time points. So that's about 3,000 or so metabolomic measurements. That's quite a lot to do. And the big challenge with this is you've got a couple of hundred women with 4 time points, and you've got over 800 metabolites. So how do you work out from that large data set which of the metabolites is actually predictive? And there's a real problem that you do the analysis and you will find something in your data set. If you do statistics on that number of samples and that number of patients, you will find something that you think might be real. So the key is not to be misled. So the study is designed in a very specific way, and because we have 4 time points, we can use the first 3 time points to look for consistent differences in metabolite levels in maternal blood. And if something is really associated with the growth restriction, then it should be consistent across all 3 samples. Not necessarily the same magnitude of difference, but it should be consistent. And if it was just a random fluctuation, i.e. nothing to do with growth restriction, then you'd have no consistency. So this is what we did. We look at the first 3 time points and look for p-values for association with growth restriction. And this is a plot of a distribution of the p-values. So p-values go from little tiny numbers to 1. And you can see the distribution here is pretty flat, except there is a skewed peak here with the small p-values. That suggests that there may be some of the metabolites in here, which have these low p-values, may be for real, but they might not. So caution is the name of the game here. So we selected the 100 metabolites out of the 837 that have the lowest p-values. So they're in this group down here. So hopefully we got the right subset. We weeded out some of them. So this rule, don't fool yourself, is kind of the key to this, because we could have just picked the top 2 and said, hey, presto, we've done it, but that would be foolish to say the least. So those 100, we then went to our 36-week sample, so the sample that we had not included in the initial analysis. And again, same logic. If something is really associated with the growth restriction, it should be different in the 4th sample, which is closest to when the sample was actually taken, when the disease occurs. So in the 36-week sample, we looked to see which of those was associated with FGR, and we imposed a pretty stringent statistical test here. So a Bonferroni corrected p-value, which comes out as 5 × 10⁻⁴. So that's a small p-value, but only using these 100. So we're trying to not fool ourselves by being very stringent. And of the 100, 22 of them passed that test. That's an internal validation, because these samples here, the 36-week samples, were not used in the initial selection of the 100 metabolites. So it's a completely different serum sample. Now, it's the same patients, but a different sample at a different time point. So that's a measure of internal validation, and that's very reassuring. So we're now down to 22. Now, some of those metabolites may well be in, or they are in fact, in the same biochemical pathways, and they are highly correlated with each other. So that's not useful for a predictive test. So we identify those that are independently predictive, and also those which improve the performance of a predictive model, because that's what we're aiming for here. We want a small number of metabolites we can measure, which give the strongest prediction of growth restriction. So having done that, we've gone from 837 down to 100, down to 22, and now down to 4. And here are the 4 metabolites plotted against gestational age. And you can see that the controls are in open circles, the FGRs are in filled circles. And this metabolite normally goes down in pregnancy. In the growth restricted, it goes down less. This one also goes down in normal pregnancy as you progress through gestation. In growth restriction, it goes down less. Converse is true for these 2. They normally rise as pregnancy progresses, but the FGRs, they rise less. That's true for both of these. So if you take the ratio of these 2, the product of these 2 divided by the product of these 2, you can come up with a predictive model. But we still have this rule: do not fool yourself, because, okay, it works with the samples we've described here, but then they're the very same samples we use to derive the model. So maybe that's just overfitting in that data set. So what we really want to do is replicate this finding in a completely different set of samples. And unfortunately, there are some different samples available. So the samples from the POP study are all collected in Cambridge. And there's a cohort of samples, a study cohort called Born in Bradford. And Bradford, as you might imagine, is not the same as Cambridge. And the demographics of the populations studied here are, you couldn't imagine things that are more different. So in POPs, in the Cambridge study, 0% of the subjects were in the most deprived quintile of population, whereas in Bradford, 38% are in the most deprived. Whereas in Cambridge, 46% were in the least deprived quintile, but in Bradford it was 5% in that top. So the level of deprivation is wildly skewed, much more deprivation in Bradford than in Cambridge. Ethnicity, very different. Cambridge cohort, 95% white, Bradford, 50% white. And the POPs cohort is very specifically and intentionally targeted at first pregnancies. That's not true in the Born in Bradford cohort. So demographically, completely different. And does it confirm it? Well, you can probably guess, because I wouldn't be telling you that if it wasn't. So if you look at the performance of the POP study initially, this is an AUC curve here. So what you're looking for is the area under this portion of the curve here. Anything below this dotted line here that's if there's no effect no benefit of the assay, you get the dotted line. Soluble fit to PLGF ratio, which we've demonstrated before in the same cohort, is somewhat predictive, but the metabolite ratio is much, much better. So that's good. And in Bradford, this is actually quite a complicated one. So this is this data here. So the POP study measured at 36 weeks has an AUC of about over 0.8. If you make the same measurement, same ratio using the 28-week samples, it's less good. And at 20 weeks, it's even less good. The Born in Bradford cohort has actually two sub-studies here, Born in Bradford 1 and 2. And those were collected at different gestation to our 36 weeks over a range of gestations. And therefore, they fall somewhere a bit below the Cambridge 28 weeks. But you can see here that this is still a surprising degree of agreement, considering the demographic difference and particularly the difference in gestational age. So we can conclude from this that this 4 metabolite ratio, which you can measure in maternal serum before, well, not necessarily before FGR, because it would be detecting FGR at 36 weeks, but that screen would allow you in principle to identify patients at high risk of stillbirth, where you might intervene to deliver the child to save its life. And the test was internally validated, so we didn't fool ourselves at that level. And it was also replicated and validated in a demographically highly distinct population. And therefore, we're pretty confident that those 4 metabolites really are informative about FGR. And finally, the acknowledgements where this work, who actually did all the work that contributed to it. So these are the folk, some from Bradford, and it's been published fairly recently in Nature Medicine. And I'll stop there. Thank you.
Excellent, and thank you all very much for the invitation and all these amazing talks so far. So this nicely carved marble funerary altar from the mid-2nd century AD, which is now incorporated into the Byzantine fortifications of Ankara, commemorates Antonia Zotica, the young wife of Annius Flavianus, a member of the staff of the provincial governor, who died aged 15 years, 5 months and 18 days on the 16th day after the birth of her first child, a son who survived her. A more modest epitaph from Salona in Dalmatia, so just outside Split in modern Croatia, too modest for a photograph, I'm afraid, was erected by Justus to his wife and fellow slave Candida. So the fact that they're both enslaved means that formally they're not legally married, but in funerary inscriptions, enslaved people generally address each other as husband and wife. And as you can see, Candida died around 30 after 4 days of painful and unsuccessful labor. Now, not many of the hundreds of thousands of surviving funerary inscriptions from the Roman Empire, and if you're wanting a map at this stage, then here is one that has most of the places I'm going to mention marked on it. And we have, as I say, hundreds of thousands of funerary inscriptions that survive from this territory, but most of them don't mention the cause of death at all, just mention the recipient. So not many of them specify death in or around childbirth. But the idea is that nonetheless, those that do clearly represent the tip of an iceberg. Now, the size of this iceberg should not be exaggerated. Popular histories and famously Lawrence Stone famously characterized childbirth as one of the most dangerous things anybody could ever do in any historical society. The one thing you should never do if you lived in history is try and give birth. But they did that without really any hard evidence. Whereas recent studies since Roger Schofield's very important 1986 essay have shown that actually maternal mortality had only really marginal impact on female life expectancy in the past, and certainly would not have generated any substantial gender gaps in survival. Having said that, maternal mortality was clearly significant, a significant feature of the Roman imperial world. One of the items I've given you on this slide is by Rachel Podd, looking at aristocratic English women, and she calculates from a collection of kind of records and other materials that the maternal mortality ratio amongst the medieval English aristocracy was about twice that of sub-Saharan Africa today. And that's the highest level that we kind of have in the WHO figures. So she would calculate that if we assume that women have between 4 and 6 children, or give birth between 4 and 6 times, that these aristocratic English women would have had about a 5% chance of dying in and around childbirth. And while Saskia Hin thinks that it's more like half that figure for the Roman Empire, without any data at all, I should say, just pretty good guesses, that's still pretty high. So she would put it at more comparable to the rates in sub-Saharan Africa, and then would mean about 2% of women dying in childbirth. That's still quite significant and important feature of the sort of demographic structure. So this is a subject that it would be really good to know more about, but where we confront, as so often in ancient history, a challenge of the absence of surviving records. So you just can't do a quantitative study. That's just not really on the cards, and therefore various people have just kind of given up. They make guesstimates and then sort of move on. But my argument here is that I think there are plenty of things that we could do, certainly to investigate, quite importantly, the sort of shape of maternal mortality, even if the precise size of the iceberg is never going to be quite clear. That we should want to do this. This is an important factor in wider studies of Roman family and social structures. So I think there are things that we can and should do then. We can and should, well, we can certainly do more with the funerary inscriptions that we have. And there are some references in literary sources. There is also a growing amount of archaeological material and more things that we can do with it. So a wider array of scientific techniques that can be brought to bear. I'm happy to talk more about this in question and answers. This slide is from one of the 82 fetal perinatal skeletons in one of the Kelis cemeteries in Egypt. And these fetal skeletons, which they've got much better at sort of aging, if you like, either buried separately or together. And the idea would be that this sort of burial, where it's clearly together with a female, would be that this is where both the child and the mother have died, probably in childbirth. So lots of things that we could do with that, but the point that I want to really emphasize here, which comes very much from the interdisciplinary context of the SRI and drawing on work that has been done and discussions in the maternal mortality thread, if you like, of the SRI, which gives somebody like me working on the Roman world an opportunity to borrow ideas and understandings from other historical time periods where you've got much better records and data, but also understandings from now, both in terms of medical science and social policy, about what the important key factors involved in determining and shaping certain sorts of patterns of maternal mortality, and indeed what works and doesn't work in respect to trying to decrease those levels and support maternal health. Now, some of those current insights are already kind of folded into some of the studies, so it's very important. The sort of first age at first birth is very important. And if you've noticed that the Roman elite have a bit of a predilection for very young first marriages on the female side. So that's something that we need to think about. Birth spacing also important. But one of the other areas that a lot of comparative work really puts the focus on is midwives, the provision of midwife services. And this is an area where the Roman Empire was really very strong. There was a distinct and well-developed profession of midwives. And we know about their practice, both from male-authored medical texts, which you have to read with some care, but also from their own representations. So this is from a tomb in Ostia, so just outside Rome, which illustrates the sort of final stages of birth. The birthing woman has been moved from the bed to a birthing chair. She's supported from behind by a female attendant, and the midwife is in front of her, helping with the delivery. And this is something that we see in various different sorts of contexts. But we also know that although midwives, obstetricaires, the Latin for midwife, obstetrics, were concentrated in the imperial capital in Italy, we found funerary inscriptions for midwives from really right across the empire. Found in cities, seems that the sort of medical culture which went with being a Roman city included some kind of midwife provision. And I've just given you a set of examples here to show both the geographical and status spread. So these are midwives that come from Rome, North Africa, northern France, that are themselves enslaved, freed. And I would certainly argue that Caelia Bonosa Massica is a free Roman citizen who kept her Roman name, but this is probably not the place to do that here. But anyway, as an example of that spread and the sort of development and entrenchment of midwife services. So this is just to say, pick up on one particular strand that I am very keen to pursue, and to return to the point that was made at the beginning about the benefits of interdisciplinarity and conversations with people working around reproduction in other fields. Thank you very much.
Okay, so from ancient Rome to mitochondria. So I'm going to talk to you about mitotherapy and how this may be beneficial for the mother and the child during adverse pregnancy. So this is a talk about cardiovascular disease, and I'm interested in cardiovascular disease because it is one of the greatest killers in the world today. And of course, even non-experts, when we think about cardiovascular disease, the first thing that comes to our mind is this gene-environment interaction. That's to say how the genetic makeup interacts with environmental lifestyle risk factors such as smoking, obesity, or a sedentary life to promote a risk of cardiovascular disease. But now, in addition to this, we know that the gene-environment interaction before birth may be just as, if perhaps not more important in setting a risk of cardiovascular disease. So that is how to say how the genetic makeup of the unborn child may interact with adverse intrauterine conditions in complicated pregnancy to set a future risk of cardiovascular disease. So this is known as the field of developmental programming of health and disease. And one of the most important consequences of complicated pregnancies is a reduction in fetal oxygenation, what we call chronic fetal hypoxia. And this is common in any pregnancy that has problems with placental blood flow, which may decrease oxygen delivery to the baby, such as in fetal growth restriction that Steve talked about, or for example, in pregnancy complicated by preeclampsia. And the important thing about hypoxic pregnancy is that it programs heart disease in the offspring. And we know this from evidence of human clinical studies which have shown that adult men and women who were born for pregnancy affected by chronic fetal hypoxia are at greater risk of cardiovascular disease. We and others have replicated this in animal models, in laboratory rats and mice, showing that hypoxic pregnancy can program heart disease in the adult offspring. But it's not all doom and gloom, because we have recently shown in a study in pregnant sheep that we can treat hypoxic pregnancy with a mitochondria-targeted drug to protect the adult offspring. So what appears to happen is that in pregnancy affected by fetal hypoxia, we generate a condition known as oxidative stress in the placenta and in the developing heart and circulation. And oxidative stress is when a normal cell is attacked by free radicals induced by hypoxic conditions, which are highly reactive and can begin to damage the cell. Now, you can imagine that if this is a developing cell in the placenta, for instance, or in the heart and in the circulation of the developing embryo or fetus, that it will express itself later as an increased risk of heart disease, for instance, an increased risk of hypertension. Now, it just happens that oxidative stress is generated within these tiny purple organelles in a normal cell called the mitochondria. And that's important because a colleague of ours, Professor Mike Murphy from the mitochondrial biology unit, has designed a mitochondria-targeted drug, a therapy called MitoQ that can combat oxidative stress specifically within the mitochondria. So in a recent study, together with Mike Murphy, we joined forces to test the hypothesis that we could blast oxidative stress in hypoxic or in pregnancy complicated by chronic fetal hypoxia. And we decided to test this hypothesis in pregnant sheep. We used sheep because they have a similar cardiovascular development to humans. So the study was very simple. We observed pregnant sheep under normoxic healthy conditions or under hypoxic pregnancy, and half of each of those groups were treated with MitoQ during their pregnancy. And then what we can do at the Babraham Center at the University of Cambridge is allow some of these pregnancies to be born and raise them as cohort of normal sheep to adulthood, because we have paddocks and grassland that can keep these cohorts for long periods. For instance, raise them until 9 months of adulthood, which in human terms is about a 25 to 30-year-old human. And of course, what we can then do is bring this adult offspring of these 4 groups to study the cardiovascular system. And what we found, or in essence, what Kim, Kirsty, and Katie found, was that adult offspring of hypoxic pregnancy, which are these black histograms, had severely higher blood pressure. So they were programmed to have hypertension in later life compared to the control group. But interestingly, that was prevented by maternal treatment with a mitochondria therapy. And if you look at the morphology, the anatomy of the major vessels, such as in the aorta, you can see that there is indices of aortic fibrosis and scarring, which are of course linked to future atherosclerosis. And again, this was elevated in adult offspring of hypoxic pregnancy relative to controls. And this again could be prevented by maternal treatment with MitoQ. So it would seem that mitotherapy is certainly protective for the heart health of the offspring. So now we began to focus on the cardiovascular health of the mother and not forget her and ask whether she's important. And of course, she is important because all the available clinical evidence suggests that women who have suffered themselves preeclampsia during pregnancy are themselves more at greater risk of cardiovascular disease. So it may seem that oxidative stress in the placenta may not only have downstream effects for the cardiovascular health of the offspring, but we think upstream effects for the cardiovascular health of the mother. And in future studies, we will be addressing whether mitotherapy in complicated pregnancy is not only protective on the offspring, but also on the mother. So if you're interested in any of what I've talked about, please email me or come and talk to me, or better still, come and join the group. This is the past and current members of our laboratory here at the Babraham Center at the University of Cambridge. Thank you.
Hello, everyone. Thank you so much for inviting me. So I'm Lou, I'm Principal Bioinformatician in the Wellcome Science Institute, specifically in the Rosers Vento Lab, where we are trying to atlasing the development of the reproductive system, one cell at a time, using single-cell genomics and imaging. So we are part of the Human Cell Atlas, which aims to create a Google Maps of the human body, and it's basically an international consortia to create a comprehensive reference map of all the human cells. And the idea is to use this to better understand human health, but also the disease counterpart. And within this consortia, so we are focused on the female reproductive tissues. So in the following slides, I will just very briefly show you our work on the adult human endometrium. So the endometrium is the lining of the uterus, and it regenerates monthly in the woman of reproductive age, and it's key for the implantation of the embryo and a successful pregnancy. So you'll understand that understanding the biology of this organ is fundamental. So here we wanted to address three main questions. So one was what are the cell types that make up the endometrium, what's the location, and how do they change through the cycle? Then we also wanted to zoom into the signaling pathways and the factors that control this regeneration. And then finally, we wanted to assess the in vitro models that we have. Specifically, we wanted to see whether the organoids are actually recapitulating the epithelial cells. So in order to answer these questions, what we did, we sequenced the transcriptome of endometrial cells from 21 healthy women in the reproductive age. And then we focus on the epithelial compartment, that's the one undergoing the regeneration. And basically, so each cell here, each dot here represents a cell from one of those women. And basically, so the message is that when we look at the epithelial cells, we were able to uncover high heterogeneity here, identifying some cell types that are well known, very established, such as the ciliated cells, of the secretory cells in the glands after ovulation, but also we were able to identify novel cell states enriched in the proliferative stage. And those were located at different regions of the lining of the endometrium. And also what was more important or interesting here is that some of the markers of those cells were genes that are aberrantly expressed in endometrial lesions, in endometriosis lesions on some cancers. And this is something we are looking at now. So next, we wanted to investigate or try to describe better how is the crosstalk between the cells in the endometrium to understand what are the signals that are pushing forward the differentiation and the regeneration. So in order to do this, we were looking at this change of ligands, receptors, and the activation of the transcription factors. And what we saw was a very striking signal for Wnt signaling in the luminal of the endometrium, while in the glands, we saw that the signal was completely opposite and biased towards not signaling. So we wanted to investigate this further. What is the role of the Wnt and not signaling in the differentiation of the epithelial lineages in the endometrium? So in order to do this, so we made use of endometrial organoids from Margarita Turco, our collaborator in the University of Cambridge, so which are 3D cultures of endometrium that are cultured in a way that mimics the menstrual cycle. And we validated that the cells resulting from this culture mimic somehow the cells that we observe in vivo. But what was more interesting is that when we cultured these organoids with NOT inhibitors or with Wnt inhibitors, we observed that we were boosting and improving the differentiation efficiency towards the ciliated lineage or the secretory lineage. And with this, I would just like to conclude that with this, we validated our model for the regeneration of the epithelial lineage in the endometrium, but also we propose a novel framework in which you can use single cell genomics and imaging to improve the models that we have now. And we are trying to repeat the same strategy in other tissues, such as the ovaries. And with this, I'd like to conclude and thanks all the members of the Evento team, especially Roser, and all our collaborators in the University of Cambridge and the Sanger Institute. And big thanks to all the donors for the consent. Thank you so much.
Thank you. I think that's a nice end of this interdisciplinary afternoon. So I'll hand over to Nick Hopwood now. Thanks very much, Antonia. So I think, well, we've really seen this afternoon that after 3 years, the SRI is in rude good health, and there are lots of interactions going on, and I hope more will come out of this afternoon. I just want to thank all of the speakers, the chairs who've done sterling work in trying to keep us to time, and especially Christina Rozek, who's organized the whole wonderful afternoon. So thank you very much. Have a great summer, and come back for more from the SRI in the autumn. Thanks very much to everyone.